Let's start our session about lymphoma in children. And before we start to work, Secretariat of the meeting asked me to announce that certificates will be ready after 3 o'clock, not after 2 o'clock, which was announced earlier, after 3 o'clock. And now we start with first speaker. It's my great pleasure to introduce you my co-chair lady, Ilske Osshlies. She will tell us about Burkitt lymphoma and new development. Please.
Thank you. And my heartiest wishes from Kiel in Germany, close to the Baltic Sea, from the Histopathology Department and Lymph Node Registry to Moscow, and to the international audience here. The topic of my talk is Burkitt lymphoma, and you know it's an important malignancy of childhood, and it's also a pioneer model of lymphoma biology in many aspects. About 3/4 of all non-Hodgkin lymphomas in childhood are Burkitt lymphomas. And the medical history of this lymphoma starts in Africa, where Dennis Burkitt in the 1916s described this tumor, these large and impressive jaw tumors in young boys, not knowing that these were lymphomas at first. And he said at that time, a curiosity can occur once, but 2 cases indicate more than curiosity. He then collected these cases and described them, and described also the enormous quick growth of these tumors, and this is also one of the major biological features of this lymphoma, with a tumor doubling time of around 2 days. The geographical distribution of these tumors in a wet and hot climate zone with a malaria endemic, pan-endemic area rose the question whether this lymphoma could be kind of viral induced. And so one stated in a Nature paper in 1962, no human tumor has yet been shown to be virus induced, but a number of virologists are now investigating the virological aspects of this tumor syndrome. And the virologists were successful. The Epstein-Barr virus was detected within cultured lymphoma cells of the Burkitt lymphoma by Yvonne Barr, a zoologist, and Anthony Epstein, a biologist. And it became quickly clear that all these endemic Burkitt lymphomas were EBV associated. And they were then more closer described as aggressively growing tumors of childhood, the median age at 7 years, and with a clear male predominance of those tumors. Predominant feature was a jaw tumor, but also kidney and lymph node involvement as well as ovary involvement was seen. And this distribution of these lymphomas was really limited to a specific climate zone endemic for arthropod-derived viral infections. It became clear with the publication of this African time of Burkitt lymphoma that this lymphoma would also arise outside Africa with the same histopathological features. And nowadays we know 3 variant subtypes of Burkitt lymphomas, the endemic and the sporadic and HIV-associated variant. And we are most familiar, or at least in Germany, we are most familiar with the sporadic Burkitt lymphoma as aggressive, mainly pediatric tumor, again, male predominance, but here in most cases involvement of the ileocecal or Waldeyer ring and variable bone marrow and CNS involvement. And only less than 10% of these lymphomas are in fact EBV associated. So looking at the definition of the diagnosis of this lymphoma nowadays, we have got the WHO defining criteria. So Burkitt lymphoma is a mature, aggressive B-cell lymphoma with a highly characteristic histomorphology for pathologists. It has a germinal center B-cell immunophenotype, so the same immunophenotype as physiological B cells in the germinal centers show, with expression of the germinal center markers CD10 and BCL6, but negativity for the anti-apoptotic protein BCL2, and a very high proliferative rate of around 100%. And certainly you all know that the MYC translocation is important. Nevertheless, no single parameter can be used as a golden standard for the diagnosis of Burkitt lymphoma. This is an example of a Burkitt lymphoma growing through the wall of the appendix. And you can see towards the left the lumen of the appendix and the transmural infiltration by these blue cells. They express CD20, a B cell marker, and you have got the close-up view with the characteristic small blue cells, intermingled starry sky macrophages that eat up apoptotic lymphoma cells. And the very high proliferative index shown with the key 67 index, and negativity for the BCL2 anti-apoptotic protein. And this picture here to the right is a photograph of a reactive germinal center, especially the dark zone. And you can see it looks very much alike the lymphoma. And therefore, pathologists very early on thought that Burkitt lymphoma might really arise from the germinal center. The hallmark translocation is the translocation 814, or variant Burkitt translocation, that juxtapose the c-MYC oncogen next to an enhancer area of the immunoglobulin heavy chain or immunoglobulin light chain, and this leads to deregulation of c-MYC. The oncogenic potential of this translocation has been shown in mouse models. MYC rearrangements are not specific for Burkitt lymphomas, but they are kind of seen in nearly all pediatric Burkitt lymphomas. We can find them in a minor proportion also in diffuse large B-cell lymphomas in children, and they are more frequently seen in a lymphoma group that is called B-cell lymphomas intermediate between diffuse large B-cell lymphomas and Burkitt lymphomas. But this entity, at least in our experience, is really rare in children and rather observed in adult. Plasmablastic lymphoma is an exceptionally rare lymphoma in children and only seen in immunocompromised children, carries 50% of MYC translocations. So do MYC translocation negative Burkitt lymphoma exist? An example, a 14-year-old European boy with a pharyngeal fast-growing tumor mass and cervical lymphadenopathy. This is the histopathology, and it really looks like a typical Burkitt lymphoma. And also the immunophenotyping shows the typical phenotype, germinal center, negativity for BCL2, high proliferative rate. Nevertheless, the investigation of the molecular translocation did not reveal any MYC translocation. Instead, in this particular case, we find a deletion of 11q24, coding for the ETS1 gene, and also multiple copies of the 11q gene. So this is not a single case. There have been publications also from Kiel, from many cases arising in children with this molecular constellation. But looking at the whole group of Burkitt lymphomas defined by molecular gene expression, these MYC translocation negative lymphomas are very rare and account to maximum 3% of all Burkitt lymphomas. The histopathology is like or similar to Burkitt lymphomas, and again, it's the pediatric age group that is affected. A limited series of cases also arising in adults have been described in Burkitt lymphomas that arise in post-transplant patients. And therefore, the revised WHO classification has now published this Burkitt-like lymphoma with 11q aberration as new provisorial category. Our diagnostic standards in pediatrics are nevertheless rather limited. If we see the clear histopathology of a Burkitt lymphoma with typical clinical scenario, we just stain CD20, CD10, BCL2, Ki67, so proliferation, and TDT, well, to exclude the lymphoblastic precursor lymphomas. We add the molecular analysis only in cases where we have troubles to be really certain that we deal with a Burkitt lymphoma. What do we know about the pathogenesis? Well, we believe that Burkitt lymphomas arises from the germinal center reaction, so from B cells that have already had antigen contact. And the gene expression profiling kind of provides more evidence for this, as the gene expression profile of Burkitt lymphomas shows a quite homogeneous B-cell lymphoma group in contrast to the other diffuse large B-cell lymphomas, and is described with upregulation of germinal center genes, and not surprisingly, upregulation of MYC and MYC target genes, and downregulation of NF-kappa B targets and MHC class I molecules. This molecular defined Burkitt lymphoma seems a quite age-independent, very homogeneous group, also in terms of prognosis and histopathology. What does c-MYC under physiological circumstances within the germinal center? It's expressed in a minor population of light zone of germinal center cells, 8%. And it's expressed in the early germinal center reaction and then repressed by BCL6 and reinduced in the late germinal center reaction. c-MYC positive cells harbor high affinity B cell receptors, and c-MYC is essential for the maintenance of the germinal center reaction. Mice without c-MYC cannot go through a germinal center reaction. This is a photograph from a fluorescence of MYC in green, and it's within the germinal center located in the light zone. What does MYC? It's a kind of very universal amplifier, rather non-specific, and acts on actively transcribed genes by binding to E-box motifs of the promoter or enhancer areas, and therefore increases the amount of RNR of the respective active genes in tumor cells. If you look at the gene expression profiling to the normal counterpart cell, then you can see that the gene expression profiling of dark zone B cells resembles Burkitt lymphomas. Or the other way around, Burkitt lymphoma cells have more or less the gene expression profile of B cells of the dark zone. So taking this together, Burkitt lymphomas resemble B cells of the dark zone. Nevertheless, they express MYC, which is rather, under physiological circumstances, expressed in the light zone of a normal germinal center. So Burkitt lymphoma might arise from a late light zone or dark zone B cell with aberrant dysregulated MYC expression. Burkitt lymphoma is much more than MYC. The translocation alone is not enough to produce a lymphoma. P53 as tumor suppressor has been described as inactivated in Burkitt lymphomas, and nowadays we know more recurrent genetic lesions of the coding genome by detection of deep genomic sequencing. About 70% of sporadic Burkitt lymphoma show deregulations of the P13K signaling pathway with mutations in the ID3 tumor suppressor gene and or the transcription factor 3, and I will show you where these act. In pure pediatric Burkitt lymphoma collectives, it has now been shown that 80% harbor this recurrent ID3 mutations. And 40% Burkitt lymphomas show cyclin D3 mutations. Where do we find these? This is a picture of a B cell with its B cell receptor, which is nothing else than an immunoglobulin. And the B cell needs tonic B cell receptor signaling for growth and for proliferation. And you can see where the transcription factor 3 acts on stimulating growth and survival and also on stimulating cycling D3, which then proposes the cell to proliferate. So we find mutations in this specific pathway, the P13K pathway, in 89% of pediatric Burkitt lymphomas along with MYC translocation. So both alterations taken together into a mouse model result in typical Burkitt lymphomas. So Sander et al. Have published a very interesting mouse model where they deregulated the MYC in the germinal center cells and also deregulated their P13K pathway. and they could show that they got tumors that had the molecular features of a Burkitt lymphoma and also the histopathological features. So in summary, we believe that Burkitt lymphomas arise from a germinal center B cell with high affinity B cell receptors, and that we have collaborating molecular events like defects of the B cell receptor signaling and proliferation pathway plus MYC deregulation, plus others that we still maybe not know. The tumor-initiating cofactors must be different in different types of Burkitt lymphomas. It might be the stimulated B cell compartment in malaria or HIV, primary immune response in the sporadic Burkitt variant in the extranodal tissues of the pia plaques, or viral oncogenes and further. The new revision of the WHO therefore states two new points to Burkitt lymphoma. One is that they add that the transcription factor 3 and ID3 mutations are seen in up to 70% of cases, including now also adult cases. And they add this new category of the Burkitt-like lymphoma with 11q alteration that I've proposed to you. So future direction will still be to understand more of the cellular and molecular pathogenesis, to better understand the initial lymphoma genesis, and to improve also the diagnostic accuracy with molecular classifiers. We would like to detect prognostic molecular markers, but more importantly, development of targeted treatments with less toxicity addressing the specific B-cell receptor-driven pathway. So my thanks to the audience, and thank you.